Hello guys, welcome back to the another session in human nervous system. Last session I have explained about nerve impulse. Where does the nerve impulse exactly occur? At axon hillock. Now, time to discuss about nerve conduction. Nerve conduction. Once action potential generated, that has to be traveled along the axon purpose to transmit from one neuron to another neuron towards their destinations. So, this is axon hillock. This is axon, entire the axon. Right? For our convenience, we have partition. This is A segment, this is B segment, so and so. This is C segment. Now, A segment, at A segment, one impulse has been generated action potential. It turns the resting membrane potential from 70 to 55, then 55 to again 70. The 55 means depolarization. Depolarization after repolarization, that is 70. After repolarization, hyperpolarization 90. After hyperpolarization, due to the sodium potassium active pump, it has been originated again resting membrane. So that A inside what has been there? Complete positive charges. When does it occurs in hyperpolarization? Outside what was there? Negative charge was there. This is the condition of hyperpolarization. When segment A is in hyperpolarization, then how about the charges of segment B? It is generally inside negative charge due to high negative protein number, and outside positive charge due to sodium and potassium ions. So it is in resting. B is in resting. C is also in resting. Let me take a little extent like this. So C is also in resting. A is in hyper membrane position, nothing but it's also called hyperpolarization. You just imagine A first undergone depolarization, depolarization to repolarization. Depolarization is 55, repolarization is 70, to hyperpolarization it is 90. When segment A is at hyperpolarization 90, that time segment B is in resting. I am telling you again, this is segmentation. We have given an understanding of our concept. Actually, that segments are not there. Then what is the length of segment if you ask me? Duration required to turn entire the polarity into reverse. That may be this much distance or this much distance or this much distance. That first depolarized segment is called segment A. Later B. B is depends upon its depolarization towards C. So whatever it is, A is under impulse, that time B is under resting. If you observe these lines I have given for understanding, actual lines were not there. Entire all this is exoplasm, right? Exoplasm, and this is extracellular fluid that is present in extracellular space. Segment B, membrane outside positive charge, inside negative charge. Segment A, inside positive charge, outside negative charge because of the process that we have seen already in the last session. Now what is happening? This positive charge comes here, then it will become positive and it will become negative. You can ask me, sir, when positive charge comes inside it, sodium gates should be open, right? That is from extracellular fluid to exoplasm. These two are two different fluids. From one different fluid to another different fluid wants to come, they require some voltage gated channels open. But here, same place. This is also exoplasm. This is also exoplasm. And this is also exoplasm. Charge is moving from same material to another material. That time there doesn't require any voltage permission. Clear? I repeat. Before you have seen movement of positive charge from outside to inside. For that, sodium gate should open. But now, positive charge is moving in the same place from one place to another place in the same region. Suppose, I would like to give an example. When you want to enter the classroom, you need a permission of a teacher. When you want to move from one bench to another bench in the same classroom, you don't need to take up a permission because it is in the same room. Whereas you are coming out of the class into it. You want to enter so that you are asking, shall I come in, sir? Means he need a permission. She need a permission to enter the class. But once you get into the class, so many students are there in the class. One student wants to move from one bench to another bench. They never ask, sir, can I move from this bench to that bench? No, why? There is no need of permission because it is in the same region. So, we can call this is local current flow. Due to the local current flow, what is happening? One charge has been come here, it will be negative, it will be positive. If this charge comes here, it is positive and it is negative. If this charge comes here, it is positive and it is negative. This charge comes here, it is positive, negative, like that. All these become negative and all these become positive. Inside the membrane, all they become positive because A segment negative charges are moved like wave. Clear? At the same time, I am explaining after this, but imagination inside and outside happen same time. And this part and this part. This positive charge comes here, it will become positive, then it will become negative. So it is also doesn't require any voltage gated channels opening. You want to go from outside to outside, no need of a permission. So that a charge is moving inside that from extracellular fluid to extracellular fluid. So that this charge comes here, this is positive and it will become negative. Now this charge comes here, so that it will be positive. All these become positive, right? And all they become negative. If you see the B, what happened? Depolarized. Now A is under resting. So it is happening because of a wave. That is depends on a phenomenon is what? Local current flow. Nerve conduction along the axon membrane occurred due to local current flow. Local current flow means the same type of substances doesn't require permission to exchange the charges. A to B, B to C. Now see what happens. This will go and it will make positive, then it will become negative. The same way, it comes here positive and it will become negative. So C will become depolarized. So and so after C, D will be same way. So this is how entire axon membrane nerve impulse is traveling by the help of local current flow is called nerve conduction. That is what the class is. Nerve conduction. Nerve conduction is helped by local current flow. Don't think that nerve conduction is takes place only inside. I said along the axon membrane. Outside also it is going, inside also going. It is wave. So that they are also called saltations. Saltation means jump. It will be three types. Suppose in case of this is one axon, in the case 1, it doesn't have any myelin sheet. This is the case 1. Unmyelinated axon. 6 neuron. In the second case, this is semi-myelinated neuron. Means run here nodes you can observe here. Like this. This is what semi-myelinated neuron. Clear? In the third case, you just see completely myelinated neuron. From axon will up to terminus, myelin sheet is completed. So this is fully myelinated neuron. Got it? In the three cases, how these impulses will travel? Observe. In first case, like this. In both sides, both the sides. So this is wave motion. Wavy. Nerve conduction is wavy. Next, the second case, if you see, here impulse is started, it wants to conduct as a wavy, but myelin sheath is made up of lipid layer. Myelin sheath is myelin sheath in chemical nature of myelin sheath is lipid. Lipid is bad conductor of electricity. What it is passing is current electricity. So that it not allowed its movement as a wavy, so that it will jump from axon hillock to first Ranvier node. Means interval between two myelin sheaths. Ranvier node doesn't have myelin sheath, means it doesn't have lipid, so it will jump here. From here to here, from here to here. Just where on the other side also. So this is what called saltations. Saltations, jumpings. How kangaroo do jumpings like that. Saltations. Saltations are happened because of incomplete myelin sheath execution. Whereas here there is no myelin sheath, so that wave. Compared to the 1, 2 is faster. 1 is fast. In case of third, it will be fastest. Why? From here to here, myelin sheath is there. There are no intervals. If intervals are there, they have to jump. So as they do not have intervals, nerve impulse reach to terminals very fastly by one single jump. So that fastest conduction or conduit. Conduit means path. Non-myelinated neurons or axons, conduction is fast. In semi-myelinated neurons, conduction is faster. In completely myelinated neurons, matured neurons, conductions are fastest. Okay, whatever local current flow is principle. This is topic of nerve conduction. Once nerve conduction happens, the purpose of nerve conduction is what? See, if this is the typical neuron structure, these are the terminals. Now, just imaginary diagram. Here impulse is started, here it is traveled, it will be any of the mode of the three, and it reached here. Once it reaches, how it will transmit to another neuron. That is called neurotransmission egonalism now. Get ready. Last concept of this class is neurotransmission. During neurotransmission, nerve impulse information is passing from one neuron to another neuron through nerve junctions. Such nerve junctions are called synapses. Let us see what is exactly. This is one neuron. They are axon terminals, also neuron terminals. And this is another neuron which is receiving the impulse from it. Okay, and the entire this junction is called synapse. For that, the junction is a duration or gap between two is called synaptic cleft. Synaptic cleft. This is presynaptic neuron. This is postsynaptic neuron. This is presynaptic neuron. This is postsynaptic neuron. And this is presynaptic membrane. This is postsynaptic membrane. Between this is called synapse, and that this called synapse. Synapse is hardly 200 angstroms distance between two neurons. Let us see. Sometimes they two together joined, there is no space. That time the impulses are passing from one neuron to another neuron directly. If the gap is there, then they need some chemical to transmit. That is called chemical synapse. So that synapses are two types: chemical and electrical. The difference between chemical synapses and electrical synapses: chemical synapses have a space that is 200 angstroms, whereas electrical synapses doesn't have. So this is belongs to only chemical synapses. Now I am explaining chemical synapses. Electrical synapses are very rare, can be found in human. So this is synaptic knob. This is called synaptic knob. This synaptic knob consists of so many vesicles. They are called synaptic vesicles. Synaptic vesicles. If you see one synaptic vesicle out, inside that a chemical is present. This is called acetylcholine. Acetylcholine short form is ACH. Acetylcholine is present inside the synaptic vesicles. Synaptic vesicles are present inside the synaptic knob. Synaptic knobs are present or belongs to presynaptic neuron only, not postsynaptic neuron. They have a receptors. Like that, they have so many receptors. As these receptors are present on postsynaptic membrane, they are also called as postsynaptic receptors. This is execution of unit of neurotransmission. Unit of neurotransmission is synapse. What type of synapse I am explaining? Chemical synapse. Now, process begin. You know well that this is the place where nerve impulse is generated, is called axon hillock. Axon hillock is the part of cytoplasm where it continued as axon. So here nerve impulse begin. Over this is nerve conduction till the end, all the six. Clear? So this is nerve conduction. Nerve impulse is first one, nerve conduction is second one in the functions in order. Third one is neurotransmission. Neurotransmission takes place here. This is third. As I discussed earlier, all the three are important functions of the neuron. Now, when impulses is traveled, you know very well how impulse is traveled because of local current flow, right? Couple of minutes back we have discussed. Because of this flow, when they comes to the synaptic nodes, due to the charge, the synaptic vesicles migrates towards the edges of the synaptic clutch. Once they comes out, due to the gradient pressure, they will be ruptured, and inside neuroacetylcholine, which is naturally called neurotransmitter. Neurotransmitter. Chemical which helps in the neurotransmission is called neurotransmitter. Example acetylcholine. So many are there. We will discuss it. Then the chemical will be released and spread and pass towards the receptor and forms a particular bridge. Neurotransmission occurred through this. As the connection is happened due to the chemical barrier, then this impulse will come and travel on this and reach. This is the impulse. Again, this impulse can generate action potential in the axon hillock. Axon hillock. It is called nerve impulse. Again, step 1, just like this. After that, again it will be passed to the nerve transmission. Again, in its end, this is nerve conduction, sorry. This is nerve conduction. Again, its end, nerve transmission occurs just like this. So, once the chemical is formed, upon the chemical, the nerve impulse is traveled. Later, this chemical can be disappeared. How does it disappear? In the synapse, some enzymes are present that are anti-neurotransmitters, nothing but anti-acetylcholine is that erase this entire path. So that on one bridge, one impulse only can travel. Another signal is coming followed by this. It has to form its own bridge again to pass from one neuron to another neuron, nothing but presynaptic neuron to postsynaptic neurons. Understand? So this is how impulses are traveled by chemical bridge. That chemical bridge is formed due to a chemical is called acetylcholine, neuroacetylcholine. On the neuroacetylcholine, if it is traveled, it can be successfully transferred from one neuron to another neuron. That impulses can be excited. But sometimes some dopamine chemical will be released. On the dopamine chemical, that entire impulses will be inhibited. That cannot be passed from one neuron to another neuron. In the journey of the impulse middle by one neuron, it will be deviated. So such impulses are called inhibitory impulses because of inhibitory transmitters. Example: dopamine, GABA, and all this. Gamma is butyric acid. Adrenaline, noradrenaline, epinephrine, norepinephrine, and acetylcholine. All these are the examples for excitatory. Excitatory neurotransmitters helps to succeed the transmission from one neuron to another neuron with nothing but presynaptic to postsynaptic. They help to transmit from one place to another place. Whereas some chemicals are there. Suppose the person who consumed the drug or tobacco, then that nicotine and dopamine, cocaine, heroin, these kind of drugs will go and bind these receptors. When these postsynaptic membrane receptors are already engaged with some drugs, this chemical cannot go and bind and fail to form the bridge. If there is no bridge, they will never transmit. That's why until they are in the effect of drug, they cannot think normally. Why? Transmissions are failed. So that they cannot think, they cannot analyze, they cannot judge the things, they cannot respond perfectly. That is why drug affects very severe on the nervous system. These kind of receptors are also present in some other, but the effect is different. Mainly when a person is consuming the drug, by nervous system is affected means the drug will go and bind to the receptors present on the postsynaptic neuron so that they inhibit neurotransmission. This is an interesting aspect. Yeah? So
typically, when you learn about neurotransmitters and synapses, you learn about excitatory neurotransmitters. Excitatory neurotransmitters will cause depolarization when the neurotransmitters reach the postsynaptic neuron. This could be enough to send an action potential down the postsynaptic neuron, but not all neurotransmitters behave this way. Inhibitory neurotransmitters have an opposite effect. They prevent the formation of action potentials in the postsynaptic neuron by hyperpolarizing the cell. This will make the membrane potential more negative, which will make it more difficult to reach threshold potential for sending an action potential. So, these inhibitory neurotransmitters are inhibiting the ability for a neuron to fire. A neuron might receive both inhibitory and excitatory information at the same time from many different neurons. It's especially common in the brain to have multiple neurons synapse with the dendrites of one neuron. Sometimes it's hundreds or even thousands. So how does the neuron know whether to send an action potential or not? The neurotransmitters have an additive effect called summation. This means the combined effects of inhibitory and excitatory neurotransmitters may or may not be enough to reach threshold potential. Sometimes one neuron will release enough neurotransmitter to cause an action potential. And sometimes many neurons will release a smaller amount of neurotransmitter, and this could lead to an action potential. The result is additive. So even if there are inhibitory neurotransmitters, it's possible an action potential could be sent as long as the threshold potential is reached. The neurotransmitters we've seen so far have all been fast-acting neurotransmitters. They can get across the synapse in less than a millisecond. and as soon as they bind to a receptor, the ion-gated channels open or close in response to the neurotransmitter immediately. This creates a very quick change in the membrane potential that won't last very long. Fast-acting neurotransmitters include the excitatory glutamate neurotransmitter and the inhibitory GABA neurotransmitter. Slow-acting neurotransmitters are also called neuromodulators. It will take hundreds of milliseconds for these neuromodulators to trigger an action in the postsynaptic cell. This is in part because they use secondary messengers to affect ion movement. These neurotransmitters are often not reabsorbed or broken down, so they spend more time in the area and can affect more neurons. Slow-acting neurotransmitters will modulate the actions of fast synaptic transmissions for relatively long periods of time, like minutes or even days. Slow-acting neurotransmitters include dopamine, serotonin, and noradrenaline. Slow-acting transmitters play a major role in memory and learning. The secondary messengers can persist for days and can lead to changes in the synapse. The neural connections are strengthened when slow-acting neurotransmitters promote an increase in postsynaptic receptors or increase the rate of ion movement through a channel. The persistent strengthening of synapses based on recent patterns of activity is called long-term potentiation. The increased activity will produce stronger, long-lasting signal transmission between the two neurons. The strengthening of synapses are currently considered to be the underlying mode of learning and memory storage.
This is, I can say, neurotransmission. Neurotransmission unit is synapse. Synapses are two types, chemical synapses and electrical synapses. And also neurotransmitters are two types, excitatory neurotransmitters, though helps in the success of neurotransmission. Inhibitory neurotransmitters like GABA and dopamine, they never help to transmit, they inhibit the impulse, so here itself neurotransmission can be stopped.
Some of the neurotransmitters released into the synapse in response to the action potential on the presynaptic plasma membrane cause excitation or depolarization in the postsynaptic membrane, and the impulse can be conveyed further along the postsynaptic neuron. These are called excitatory transmitters. Acetylcholine and noradrenaline act in this way, driving the response of muscles or glands. Conversely, other neurotransmitters, such as glycine and gamma-aminobutyric acid, GABA, hyperpolarize the postsynaptic plasma membrane. This in turn reduces the transmission of impulses and the activity of the postsynaptic neurons. These are called inhibitory transmitters. For example, the binding of GABA to its receptors opens up the chloride ion channels. This results in an inflow of chloride ions and hyperpolarization of the postsynaptic neurons.
So further transmission cannot be occurred. So this is the part. By this I can say nerve functions 3 can be done completely. One is nerve impulse, in the last session we have seen. Next, nerve conduction, before this concept I explained, and neurotransmission, this is the one. All the three are very important. Upon that, definitely one question will be there in the main unit examinations. Hope you understand. Revise with good notes. All the best. Thank you. And next class I will start human brain. Bye.